Hello, sir.
Hi. Okay, so now let's start. I was waiting for everyone to get here. So we haven't met in 2 weeks. We had some difficulties last week with scheduling. So I think Ujjwal, Asmit, and Cross had a term paper they had to do. How did that come out for you?
Sir, can you repeat, please?
Oh, I said you guys had a term paper due last week. How did that turn out? Did it go well?
Yes, sir, it was well. It
Oh,
was fine.
good. Yeah, good. Um, so you guys have been working on I guess, the digital Bacillaria stuff. You've been working on a lot of analysis and writing up like methods and things like that. So where are you guys right now on this?
Oh, so we have started like we have put the frame versus like how many boxes, like how many bacillus cells like we are able to identify. We have plotted that graph and we have taken like approximately 93 to 95 frames which are continuous in nature, and we have tried to extract the position of bacillus in these frames through the coordinates which we got of boxes, and we are trying to figure it out like how they are changing. We are working on that pattern as well, and on the documentation we have started documentation. We are about to complete it, like there are some final touch-ups which are left, like improving grammar and making it like as a researcher I asked, like we have to make it more and more mm, uh, more and more compatible so that it can be good for the person who is not so much familiar with neural network as well. He can also read and understand that. So we have started that as well.
Okay.
And we should upload the documents in the shared you have on the doc you have shared with us by maybe Sunday or coming Monday.
Okay, yeah, so you did get the link to the document that I'm starting, the Google Doc.
Yes, sir.
Okay, so yeah, so Richard wanted to share that. He wanted to have like a common Google Doc for everyone to contribute to. He likes to work with Google Docs, and it's probably better for writing papers. Although I also like to do it in GitHub, but I think in this case, Google Doc is better to collaborate on because you can do a lot of things of formatting in Google Doc, which is pretty advantageous when you get down the line for like references and things. Um, if our what you could do is just like you could work directly in the Google pub or in the Google Doc that we shared. You could just put in what you have now. If you look at the Google Doc, I've just made it so that there are a lot of empty spaces, and so you could put it in. I guess put some in the methods and put some in the results, and then we'll go through it maybe next week, and then we'll figure out because things will get shifted around anyways. So it's probably better just to work from the Google Doc at this point. Yeah, even if you don't really think it's ready, that's fine. Just put what you have in there, and then we'll work from that document. The reason being is because I want a little bit more interactivity between like myself and Richard, and it does help that if you have like someone like Richard who's biologist by training and also a physicist, but he has like a lot of expertise in Bacillaria for him to read through it and say, I don't understand this thing about machine learning. So I think it's better. I think it's beneficial to have it like in the public document. And then in terms of your uh, results, they sound good. Do you have anything that you can show or?
So actually, I have to connect from another device to show that, but I can share it over Slack with you today.
Okay, yeah, we can put it on Slack and share it. Um, so that's good. And then you're gonna make like a pull request this come in the next few days, or
So, as you have said, I will update the document within 1 or 2 days,
Okay.
whatever we have got, and I will share the graph which we have plot, like as the Richard said, I've asked over Slack. And once we are able to figure out something from XML files, we will start sharing the results over Slack with you.
Okay. Sounds good. Um, so Paras and Asmit, do you have anything to add to that?
No, I guess till now we have trained on 90 or some images and corrected the coordinates of the boxes. So we'll be plotting them and like saying if we can get something useful from that, and maybe uploading them in the coming few days.
Okay. All right. Paras, do you have anything to add?
No, sir, that's pretty much it.
Okay. Well, we'll go through the issues later, and we'll put some things on the board, and then. But I wanted to then turn to Vinay and see how he's doing. So, what's the update from last week?
Hi, sir.
Hi.
So, we're still in this discussion of what might be a good thing to do, right, sir?
Yeah.
So, last week we had some chat. So, I like to if I can get a clear view of the inputs and outputs of the software, then I think I'll be good to go. So, I have a few doubts regarding that.
Okay.
And if you can open our chats, like our Slack chat, I think we can navigate on that. Like we can build up on that.
Like the chat that we were having, or different? Okay. Let's see. Paras has a request. What was the request that you had? You have a paper due today, so you want to just do your. Okay. So you want to leave early, or? I mean, we can do the
Me?
board real quick. Oh, I'm talking
Yes, sir.
to Paras. Sorry.
Okay, sorry.
Yes, sir. We wanted to finish our stuff first.
Okay, so why don't we just move to the board and then I'll go to Vinay. So sorry, Vinay, I'll be with you in a minute. But
It's okay, sir.
Okay, I want to go to the board here. So digital Bacillaria, and if you go to that link, you go to the projects link, and then you go to the board. Why don't I share my screen with you for this so I can. Okay. Can you see my screen? Okay. So this is the board, of course, in progress and to do. So let's go through the things real quick, and then we'll add on what we need. So number 34, that's Paras. He's still going to give a presentation at some point. Is that something you still want to do? Hello? Can you still hear me? Everyone's muted.
Yes, sir. Yes, sir.
Okay. Paras, can you, do you still want to do number 34? What about number 27, mathematical model development? Where are we on this issue? Is this still in progress?
So yes, we have kind of extracted the coordinates of the Bacillaria image in 93 to 95 continuous frames, and we are trying to figure out like how they are moving. And so yes, we have made an improvement in this.
Okay.
So,
Uh, Okay, well, give me some updates on that as you're going along. You know, I mean, even if you don't have any questions, just kind of like running things by me, and then I can, maybe I'll get Dick in the loop on it if it's something that sounds like it's a problem or something it sounds like we can add to in some.
Yes, so like we have what we are planning to do is like we have the four coordinates of Bacillaria bacteria like Xmin, Xmax, Ymin, Ymax, and we are trying to figure out like how they are moving. So what we are doing is we are trying to find the difference between these four coordinates in the 93 continuous frame so that we have the displacement like something. So after we get the displacement in these frames, I think like we can calculate the velocity and all these things. But the problem which we are facing right now is like in the initial frames, like around 43 to 47 frames, the values are not changing much. Like they are getting wider rather than moving somewhere. So like, should we consider getting wider as movement as well, or like it is some other purpose?
Getting wider. You mean like the actual like filaments are getting wider?
It is kind of yeah, it is kind of like stretching itself. Like we are not able to understand like what is there. It is kind of stretching itself.
Oh, like it's stretching outward or yeah, I think that's part of the movement though. I mean, like they're going to be like different components of the movement, so it's not just going to be like maybe a smooth path. There might be some like moving in one direction, but it's kind of drifting. Because remember, we had the issue with there. Of course, the videos aren't like excellent quality. I mean, they're not like real easy to track. But then you have like the thing is actually pulsating and kind of like moving a little bit, to keep itself maybe in a certain position. So that I would consider that to be a component of the movement. So yeah, that would be. I don't know how you want to deal with that, but that's part of the movement.
So okay, so like so we will like by now we are trying to get the movement like upward, downward, and rightward, leftward. Like we will now from now we will start considering this as a movement also.
Yeah, yeah. I mean, you could break it into different components. Like you could say, this is like the wideness, and then we could figure out what that is later, and then you could figure out like a trajectory, which is like the main component of the movement. So they're going to be different. Like they're going to be different parts of it.
Okay, sir.
So I mean that's yeah. Just we should have more. I think we should discuss that a bit more, like in Slack or whatever, just to make sure that we know, kind of like that we're on the same page about it. It helps us all think about it more. And then we have so description of neural networks for approach for biologists. That's still outstanding. That's sort of my responsibility. I haven't done that yet. Or well, I guess we talked about that a little bit with Asmit and Ujjwal. So you can address that when you're ready. The template methods, I think that's what we're working on with the methods. Like I said, that can go in the Google Doc, and we can get that into shape there. Documentation or procedure, the same thing. It might take a couple of rounds of edits to get it really nice and readable. Then reviews Ubuntu, that's that paper. That's sort of, I'll put that in done because I think that's just kind of like something that I asked you to look at, but it's I don't know how relevant it is to this. Then the model evaluation, that's something that we're kind of still going on about. We're maybe not quite there yet, but we're thinking about it. Then I need to go back to number 34. I think Paras dropped off the call. But is this something that you're still interested in doing the presentation to the group? Oh, let's see. Let me put it in the chat. Okay. And then this is issue number 34. Oh, okay, for the presentation, I've read some papers. Okay, so pick a paper and create a presentation.
Is everyone? Hello?
Hello, yes.
Hello?
Yes.
Yes, so that about the presentation, I have read some papers, but that are of the area of NLTK, neural, natural processing. So is it okay is that if I present paper about other domains?
Oh, that's fine. Yeah, that's a good, just whatever you want to present on, as long as you know it's in the machine learning area or what you're talking about. So, yeah, it would be like 15 to 20 minutes. And then, we'll do the 15 to 20 minutes in PowerPoint slides, and then people would ask questions about the paper. About, I don't know if you can answer them because sometimes they're very opaque in the paper about how they do things, but we can discuss it.
So I think let's just move this issue into the future development, and I'll let you know when I'm pretty sure about the paper.
Okay.
So because of deadlines and exams, I couldn't find a good paper that I can present that I know very much about it.
Okay. Well, yeah. Whenever you're ready, you can let me know, and we can do it.
Okay.
Okay. So then, let's see. Number 39. We define output parameters. Yeah. An output is CSV format. So I think this is
This is done, sir.
What was that?
This is done, sir. Like we have extracted that single information into CSV for analysis.
Okay, yeah, that's the XML. Okay, we'll put that done then. And then need to work with output data. I think that was something that we didn't do yet. We've just been busy and we haven't been able to get the meeting going. But do you think you'd benefit from having like a session where we just look at the output data, or is that something that we can do over Slack?
I think so. Like we can do it over Slack, and once like we are very close to getting what is required, and we can have a meeting for that.
Yes, so over Slack would be better, I think, right now because.
Okay. Yeah. Okay. Well, we'll do it in Slack. And it's just a matter of like maybe sharing plots and like showing how what the data look like. You were talking about some of these things that you're having a little bit of problem with. Just putting that out and like I'll give me my feedback on it. So that's good. We'll put that in progress. So we can put it on Slack. I guess this week you said so. We'll be doing that. We'll just put a note in the issue. We'll say we'll do this over Slack. Alright, and then we don't necessarily put a time thing on it, but let me get out of this. Okay. And then, okay, so that's done. Let's see. Model features within and between frames. We're kind of doing that now, so that's kind of in progress. Create video resources for Bacillaria biology, that's still in progress. That's my thing. And then define features within and between frames. And that's kind of what we're doing as well with the modeling, with the min-max modeling and the displacement. So yeah, I look forward to seeing your stuff on Slack, and we can discuss it there this coming week. Now, are there any issues we need to put on the board for the coming week?
These are the, I guess, very large and broad issues, and I think like it covers most of the things like we are doing right now.
Okay. All right, I just put a new issue, put materials into Google Doc. So that's something that is to do this week. And then I'll put everyone on that. Here. Visual on it. Go. Go back to 100. And now, any other issues? No, sir? Okay, thank you. So, I think you guys are done for today. If you want to work on your paper, that's fine. Thanks for the update. I look forward to seeing you in Slack this week. If you want to stick around for Vinay's update, you can do so.
Okay, thank you, sir.
Okay, you're welcome. Thank you, Vinay. So, what's your update for this week?
Yeah, sir. So we've been discussing about the cell tagging software, like what will be the requirements, what will be the inputs, and how a user can use the software, right?
Yeah.
So, like, I have a little bit of small doubts regarding that. So, like, if you can open our Slack chat, I think we can build up on that conversation.
All right. I'll have to open it up on a different device, but I can do that.
Sir?
Oh yeah, I'll open it up on a different device. I can't open it up
Okay.
on here. Yeah. It's like I'm already having problems with memory on this. So we discussed the self tagging software. But was this the thing? So this was the trying to find your place here.
Or I can post our conversation in the chat here so we can discuss
You wanna?
it here.
Can you share your screen if you have the chat up on your machine?
Yeah, definitely. Yeah.
Okay,
Sure.
go ahead. Okay, there we go.
You can see my screen, right?
Oh yeah, I can see your screen.
I was talking about this, like our chat in the last week. So, like when we spoke about how the software should be, like what will be the inputs and what my work will be, and what's the desirable output, and how a user can interact with our software. So, regarding this conversation,
Yeah.
uh, uh, I had a little doubt. So you're referring to inputs would be an image and perhaps a metadata file. Do you mean by metadata file it's the CSV file?
Well, the CSV, I was thinking about that. I wasn't very clear on this answer because I was thinking about it as I was writing. So I think maybe a good way to think about it would be the CSV file might be like a reference file. Like, so the positions and the names should kind of be there. Like, you know, on the server. And then what people would upload would be like images. And what I mean by metadata is like the name of the movie, like where it was captured, what the stages are of the movie, and things like that. So like
Okay.
yeah. So the actual CSV files, like the actual cell positions and the names, I think those would be good if those were just kind of like already in place. And then when someone uploaded a video, you know, you take the each frame and you'd figure out where the midpoint of the image is, and then you'd figure out like in a Z transform, like I was telling you, with the position, and then kind of find the closest match. That would be the and then the metadata would be something that you could attach to that result, and and say like this is what this represents.
So I want to repeat what I understood, like so you can point out if I misunderstood something.
Okay.
Our software will be a web app where in the server we'll be having a CSV file, like the data file like you gave me, like we discussed about the CSV file which I have, which has the XY coordinates and XYZ coordinates, and the cell names and the timing at which the cell is born,
Yeah.
right?
Yeah.
So whenever a new user comes and he or she uploads a video, since this uh, since the C elegance, like they for suppose the time at which a cell bonds is roughly at the same time. So with our reference CSV data, we can try and label, we can try and tag the video which the user has uploaded, right?
Yes.
So like for suppose, let's say we have in our reference data, let's say we have AB cell will be born at a 42nd minute.
Yes.
So we can split the 42nd frame, or some frames in that time frame, and we can try to predict where the AB cell will be in the new user, and we can mark that cell, and we can again put it back into the video and let the user download the video, right?
Yeah, that would be good. Or maybe, well, you could, I guess you could you do it that way? Could you do like could you actually like take the frame, mark it, and then reassemble the video on a web server? Or would it make more sense to have like a file that you could download with like all the cells identified with their centroids and their sizes? Like you could have, you would upload a video, and then you would download like an analysis. It would just be like in a CSV file. So it would be like one column would be the name of a cell, then the prediction of where that cell is in the image. So it'd actually be you're just taking like an image, and you'd take each cell, you'd segment the thing, and then using the data in the CSV files that we have, you would predict what each blob is, like in name of the cell, based on the location. It would be a prediction, and then you would have this. You could report the size of the blob, the size of the cell that you're predicting, and then the centroid position of that prediction. So like it would be a prediction, so people wouldn't think that it was like an absolute answer, because that's the way they do it in, say, like bioinformatics. They make a prediction based on the data that they reference data that they have. So would be a prediction of what all the cells in that video, what they are, and the time that's supposed to be happening, and the size of the cell, and then the centroid position.
Yeah, sure, sir. Now, now I think I have an overall idea of where this project is going. So everything seems fine. I think it will be great if we can do this, but I'm only concerned about the segmentation part, because the segmentation that we did, I'm a bit concerned about how we can get the centroids and how accurate would that be. So that's the only concern I have. Apart from that, if we can get the centroids from a segmented image, like let's say let's come from the beginning. Let's say our user uploaded a video of a C. elegans embryogenesis process.
Yeah.
And what we can do is that we can split the video into frames, and we can segment each of the frame, and for each frame we can calculate the centroids of each of the cell that are segmented, like in our segmented image.
Yeah.
So based on those points, we can based on those coordinates that we get, we can try and match with the reference CSV data. Like let's say we detected a point at some 100 at the x coordinate and 200 y coordinate, and we'll try to match. We'll try to find the nearest point in the reference reference data, so that we can think that, oh, that and this must be the same, and we can automatically assign that tag to this centroid that we found from the segmented image, right?
Yeah, yeah, I think that's.
Yeah, yeah.
And then you could use, like, I guess you could use one method to do it, or you could give people the option of using two or three methods. The reason being is that if one method is very bad at a certain video, you might get
Okay.
another method that works a lot better. So, like, again, it's like the bioinformatics example. You have what they do in a lot of, like, when they predict gene sequences, they'll use a couple of different options for, like, algorithms, and then they'll put the sequence in and they'll do a match, and then they'll get a certain, like, accuracy rate, and maybe report the accuracy rate of the match, if that's possible, or some way to know whether that's a really good fit to the data or not. But then they might use another algorithm to get a better result. So I mean, if you've looked at the digital Bacillaria stuff, these Bacillaria are these microorganisms that swim around and they move around a lot, and so one algorithm might be great at, like, capturing one aspect of the cells, but maybe not so great at capturing another aspect when they're moving around. Then it might be a bit different. So that's a problem maybe in some of the input videos if people are uploading things that aren't, like, super, controlled, in their imaging. Because when people collect images, sometimes, they don't mount the specimen as well as, like, some of the videos that we've been working with, because those are from publications. So I mean, there is maybe a little bit of slop in the images that maybe one algorithm is good at, but another one is not.
Yeah, 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 yes. So, what I understand from that is that, like, from the segmentation part, I think we can work on it a bit, and we can adjust it neatly so that we get the centroids as accurate as possible, because the tagging of the cell mainly depends on the centroids that we get from the segmentation. So reducing that error greatly reduces the overall error. I think that greatly influences the overall error. So I think we can work a bit more on the segmentation part and ensure that each cell is neatly divided instead of a noisy thing that we are getting right now.
Yeah.
So once we get that, and then if we are able to like get some clear boundaries, and then if we can calculate the centroids of each of the cell from the segmentation maps, then I think we can we can just do a closest point search between the calculated centroids and between the reference points.
Yeah, I think that's a good strategy. Yeah,
Yes, sir.
to have some measure. Actually, it's good to have some measure of, like, yeah, it would be like kind of like almost like a measure of accuracy in a way, but
Yeah, how good we are able to do this, right?
yeah, yeah.
Yes. So I'll think about that measure. So currently, I think what's urgent is to work on the like refining the segmentation, and once where we are satisfied with the segmentation, and then work with the calculation of centroids, and then it's an easy. I think after that it's a bit easier to do, because it's just the closest match between this reference points, and for the metric on how good this, like, which helps us estimate how good the software is, we can just verify on several new maybe a new videos, and for the first time I think we can do it manually, like uh, uh, check how good the software is, and then we can try to derive some mathematical formula which gives us a metric, and which in turn helps us find out how good our software is.
Yeah, I think that's a good way to go about it.
Yes, sir.
Yeah. Okay, yeah, is that,
Uh,
so that's pretty much the
yeah, I think
jury of
that.
doubt
Yes,
solved.
yes,
Yeah.
yes, sir.
Okay. Well, why don't we go to the board?
Yeah,
Let
sure.
me see. I'll give you the link again.
I have it open.
Oh, okay, good. So we'll just do this. So let's see. Let's do the in progress and then to do.
Yes, sir.
So uploading documentation for unsupervised approach number 32. Is that in progress still?
I think I've uploaded the PR. Like you merged the PR this week, right?
Yeah, yeah, it was
So
in
in
that.
that, I wrote the complete like I've divided the documentation into two parts. Like one is for the users and one is for the developers. Like the users section mainly focuses on like it's an abstract on how the entire software works. I've given steps on how to get the jar file and how to install the image and get and what are the steps to basically get that working.
Okay.
So, and for the developer section, I've added guidelines on how to set up the environment first, like how to get familiar with the workflow of writing plugins for image. Like what I've included some links which really helped me when I was doing the work, and yeah, I think that is done for now.
Okay, good. Put it in done. Get MATLAB code running inside of Python web apps.
Yeah, we spoke about it, and they had a condition that like they had a MATLAB plugin for self-star algorithm. It was mentioned that we need MATLAB enterprise version for that.
Yeah, that's what I
Like
thought.
we need to, so I think that's a block, and we try to like uh, go away from the self-star algorithm and focus on the self-tagging software, building it ourselves.
Okay,
So I
I'll
think we can remove that issue.
yeah, put that in done. I mean, there was just one way to go, but
Yeah.
um,
Yes, sir.
implementation of plugin development, scheduling additional time for enhancement. I
Yeah,
think
I think
that's
I think what we tried to avoid in the beginning that came to be the most important one because
yes,
the time for the scheduling time for enhancement. I've asked it because of this only. Like I felt that segmentation. I can maybe we can put some more time on it and see if we are able to get it done, like get it more refined. So I think we are back to it again, and this is our main issue right now.
I will still, yeah, leave it open.
It's still progress. Yeah.
Okay. Implement a method for identifying noisy outlier regions, number 27.
I think this 27 and 16 are linked issues because if we are able to detect the outliers, that in turn means that we are enhancing the segmentation. So even that should be in progress, I think.
Alright. I'll just put a note in here real quick. Um, segmenting bright field images.
Like we wanted to focus on this cell tagging software instead of this, right?
Yeah.
So what do you think we should do with that?
Well, we could probably just put it back into do, and if it doesn't get done, it's fine. You might actually even use these bright field images to test the thing that you're talking about building. So, like, once you have the
Okay.
videos, then you just see how those work. And then they might not work very well, but we at least then know what kinds of videos work and which don't. So, put that back into do.
Yeah.
Work on labeling of cells at different stages. That's
Yeah, we
probably what
can.
we've got going right now with the
Yeah,
CSV
pull that into
files.
in progress.
Okay. Yeah, I did put that in there. Contingent on CSV file. So, yeah. Work with web app for cell star segmentation.
We can. I think we can rename that into like we can rename the cell star into cell tagging software, and we can pull that into in progress.
Uh, Progress here. Okay, I gotta finish up here. The DevoWorm and Neuroinformatics outreach that's in progress. That's to do.
Sir, I have something to tell about it. So,
Okay.
in the in the past 2 days or 3 days, so I received a mail from Malin ma'am asking all the GSOC students to tell her if we are interested in doing an outreach. Like we have a scholarship, like $500 scholarship for students who like to go to conferences or to go to travel or like travel to conferences for the sake of community outreach, like presenting our work or something like that.
Okay.
So she asked us to put a proposal for that. So maybe I'm thinking about it. I'm thinking to put a proposal about my work, like what I've done in the past 3 months, like and and like spreading the word about like intersection between the neuroscience, computer vision, and also bioinformatics. So and we can. So I was thinking about that. But like in the past week, like we I had been busy with all like so much stuff is happening right now.
Yeah.
Like we're also I wasn't able to write up the proposal. So I think I'm thinking of writing a proposal in a 2 or 3 days, and I'll send her and see what will happen.
Oh, that's good. Yeah. Sounds good.
So,
Yeah. Okay. Well, we can, I guess, put that here, but keep me in the loop on that.
yeah, yes, sir. After I write the proposal, can you do a review on that? Like,
Oh yeah, yeah, I
what
will.
what points do I need to add? Like, if I'm missing something important, or how to make it even better, something like that.
Oh sure, yeah, I'll do that.
Yeah,
Okay,
yes, sir.
great. Yeah.
So, yeah,
So let's finish up here on the board. Begin to work on semi-supervised approach. That's sort of what we've been
to
talking about.
do.
I'll put that in progress. And then I put one in progress here. Data for centroid positions and labels. That's in progress. I still have to get
Yes,
you the ones for the terminally differentiated cells, but I'll get that to you later today.
yes, sir. Sure.
Okay, so well, thanks for meeting, and we'll work on this throughout the week. You can message me on Slack, and I look forward to talking with you, and we'll see you next week.
Yes, sir.
If you have any questions about the stuff you were telling me about, let me know on Slack. So have a
Yeah,
good week.
I had pretty much cleared up in this meeting. Like I had some doubts regarding that concern me. In this meeting, I think I now have a clear view of what's ahead.
Okay, sounds good.
Yes, sir.
Okay.
Thank you.
All right, talk to you later. All right, bye.